Good morning, good afternoon, and good evening to all of our webinar participants, and welcome to the second webinar in our series on microplastics and health. My name is Margaret Spring. I'm the Chief Conservation and Science Officer at the Monterey Bay Aquarium, and I'm the chair of the planning committee for this webinar series. Information about the entire series, including the full planning committee roster, can be found on our website, and that'll be in the chat if you can see that. The series is an activity of the Environmental Health Matters Initiative with the acronym EHMI, which nobody probably knows, but that's what we refer to it as, a forum for the National Academies of Sciences, Engineering, and Medicine. This series of webinars intends to provide a solid baseline of information and knowledge to inform future work and decision-making on microplastics. More specifically, the series aims to assist in identifying research gaps and regulatory needs and promote cross-sector dialogue on decision-making regarding microplastics and their potential impacts on human health. So on behalf of the Environmental Health Matters Initiative and in collaboration with the National Academy's Roundtable on Plastic, I'd like to welcome you to the second webinar in this series that will focus on distribution of microplastics in the environment and human exposure pathways. Looking back, during our first webinar on February 27th, our speakers and panelists covered methods for sampling and characterization as a foundational capacity to support exposure assessment and hazard evaluation as we seek to assess potential human health risks. And I encourage you to watch it. It's on our website and recorded. During the first webinar, the speakers identified the importance of proper sampling, extraction, and analysis methods, considering matrix effects that may impact material identification, and they highlighted the need for QA and QC, including blanks, controls, and adherence to ISO principles for method transparency across different media, which means water, air, soil, and biological samples. They also discussed how effective evaluation requires collaboration to properly design collection, analysis, and toxicological studies, leveraging past learnings from nanomaterials to improve characterization and risk assessment. And finally, they pointed out that a centralized microplastics research database and continued development of ISO and ASTM standards are essential for data sharing, standardization, and informed decision-making. So today, the goal of this webinar, our second one, is to promote awareness and provide a standard baseline understanding of microplastics distribution in the environment and the most prevalent sources of human exposure. Future webinars, we have two more, will build on this as we address topics around potential hazards in human health and complexity of assessing risks, and the considerations for risk communication and exposure mitigation. That's a lot to cover in this short series, but we aim to provide an effective baseline of information to inform future research and discussions. The committee would like to emphasize that the content presented in this webinar is not intended to represent the formal perspectives or recommendations of the National Academies. All views and opinions expressed are solely those of our volunteer guest contributors. This workshop series aims to bring together a variety of stakeholders, experts from different sectors with diverse expertise and experiences. For this workshop, we want to thank the Planning Committee and members of the Roundtable on Plastics for their thoughtful contributions, the National Academy staff, of course, and today's speakers and panelists, all of whom have worked hard to bring together this exciting event. So now let's turn to today's webinar. We'll have two sections in this webinar. The first will be two presentations to set the stage, followed by a short Q&A. Then we'll turn to a second session, which will be a multi-sector panel discussion that will delve a bit deeper into some specific topics. So audience members, you're part of this too. So you're invited to submit your questions at any time using Slido. Even if you do not have a question, still go to Slido where anyone can upvote questions they want answered. The link for Slido can be found on our website and in the chat. And with that, let's get started. Our first session, as I said, the speakers will provide an overview of the sources, types, and distribution of microplastics in the environment and the human body, as well as how to deal with exposure data in the context of effect and risk assessment. It's my real pleasure to introduce our two esteemed speakers, Dr. Tracy Woodruff, who's a professor in the Department of Obstetrics, Gynecology, and Reproductive Sciences and Philip R. Lee Institute for Health Policy Studies at the University of California, San Francisco, and Dr. Bart Colemans, who leads the aquatic ecology and water quality at the Wageningen University. So all biographies of our speakers can be found in the agenda on our website. And each speaker is going to have 12 minutes to provide their thoughts, and then we will open up the session to questions. And as a reminder, audience members can submit or upvote questions at any time using Slido. So with that, Tracy, please unmute yourself and start sharing your screen. See if we can.
All right, I am already unmuted.
All right, well, we'll turn it over to you now. Thank you.
All right, well, thank you. And thank you everyone for joining this really excellent webinar. I'm going to set the stage for the broad overview about sources and exposure, and then Bart will be following up with some of the details on microplastics and exposures. So first, I want to say that I have okay, I have no financial conflicts to declare. And as Margaret mentioned, I am from UCSF. I run the program on reproductive health and the environment and the Earth Center. And I'm going to be speaking from some work we did with the California State Policy Evidence Consortium from the University of California, and a report that we did, which is referenced here on microplastics, the health effects of microplastics and microplastics policy that was requested by the state of California, and the links are in the references provided for this webinar. So the context of the microplastics and plastic exposure is the ever-growing increase in plastic production, which has doubled since 2008 and is projected to triple in the next 30 years, which means this is the context for understanding the extent to which we have exposures, how they may be increasing over time and could increase over time. And just to set the stage for where plastics are coming from, for example, in the United States, plastics are primarily made from frac gas and chemicals, and they consist of two primary types, those that are primary microplastics, so they're manufactured microplastics, they're used in a variety of different products. And then there's a wide variety of microplastics that come from degradation of primary materials, which we will be talking more about in this brief time that we have together. And the degradation of these materials leads to human health exposures. So I think I put this in because I wanted to also talk about the 7 common types of plastic polymers that are used in production globally, and that's because that has relevance to the types of microplastics that people are measuring, both in terms of exposures and in people's bodies. They're listed here. I'm just going to highlight the polyethylene terephtalates and the polycarbonates because they comprise the highest percentage of use in plastic production, along with polypropylene. And so some of the studies that have been coming out attempt to disaggregate the microplastics they measure in people by these different types of plastics. So plastic fragments, we are exposed to plastic fragments because they are entering into the environment. So these plastics, primary plastics, which are produced from fossil fuel chemicals or fossil fuel-based plastics, they break into small fragments and including microplastics, and they enable long-range transport, so they can they're found in many locations. That's lots of reports about how they've been measured in the ocean, and because they're getting into these different locations, that presents opportunities for exposures to us. I know you probably talked about this in the first webinar, but I think it's really useful to emphasize that these microplastics, they're really small, particularly the ones that break down, and the microplastics that get out break down into even smaller plastics, and they're mostly invisible to the human eye, which is why the first webinar was so important to talk about this. It represents challenges in how to measure them. I think the other important component of thinking about microplastics and exposures is that microplastics are used not only in single-use plastic and plastic packaging and plastic materials, but I think, and this was eye-opening for myself working in this area starting several years ago, is that they also, certainly packaging is the largest source of plastic waste by industry, but also textiles, so clothing made from plastics. Other sectors, including tire wear, so that means tire wear and is an important source of microplastics in the air, and then other consumer and institutional products. And I also wanted to point out that you can see that based on the different type where the microplastics are coming from, they look different. So these are some images from microplastics measured from these different sources. And then that's going to be also important when we're thinking about exposures. Bart is going to talk more in depth about this slide, but essentially, because you have these large plastics, they break down into the environment. They represent a source of microplastics that are in waters, in air, and those can migrate to get into food sources. So for example, they can become bioavailable to fish. And then when we consume the fish, then we are then consuming the microplastics that are in the fish or in other animals. Also, I want to note that microplastics can be part of food packaging. You can get exposures to microplastics through food packaging. It gets into water, so you can get it through drinking water. So we are exposed to microplastics through a variety of mechanisms, and Bart is going to talk about this a little bit more. He's also going to talk more about this, but I just want to preview that this comes from a very nice article that he did in conjunction with other researchers in science. And it talks about, because there are all these different sources of microplastics and they are in the different medium to which we are exposed, air, food, drinking water, and dust, we therefore, a lot of studies have been now coming out measuring microplastics in many different parts of the body, including feces, placenta, breast milk, blood, and there's some recent reports of microplastics measured in brain tissue and testicles. Now, I want to point out a component of this that has not been well covered in a lot of this research, and that is that certain groups or certain people in the population are going to have higher exposures, and in particular children, because of their behavioral patterns that put them in contact with, can put them in more contact with microplastics. And that's because they have behaviors like they crawl around on the ground, they put their hands in their mouth a lot. And I wanted to highlight that this study where microplastics have been measured in dust in the home. This is from a study published in Environment International showing the distribution of two different types of microplastics. If you remember from the slide, these different polycarbonates, PET and PC-based microplastics that were found in indoor dust in every location where the indoor dust was collected globally, and that you can see that in this particular study, most of the composition of the microplastics found comes from PET. But the important part is that they also estimated, if you're finding this in dust, you can use EPA tools to estimate what the exposures are. And while a lot of studies in terms of measuring microplastics in people have focused primarily on adults, you can see that if the estimates are, though, that children, particularly toddlers, would have much higher exposure to microplastics due to the fact that they're crawling around on the floor, putting their hands in their mouth and coming into contact with dust. And so I think this brings up, and I'm going to conclude, and a really important factor when we're thinking about how to characterize exposures and thinking about the health effects is that we know that microplastics are ubiquitous in the environment and they're measured in humans. I also want to note that they're persistent and bioaccumulative and only break down into smaller particles, and they can take, well, they can never leave, but they can take many, many years before they're gone. And that what we see typically with environmental exposure studies in general is that as increasing production goes up, or if you decrease production of chemicals, that can lead to either increases or decreases in human exposure. And with increasing plastic production, that would mean increasing exposures. And that we really need to think about exposure studies that are focused on understanding those that have the highest exposures, including children's and infants. So the recommendations are to focus on that. A lot of the studies have focused mostly on adult exposures. So we need to look at developmental stages. We need more tools to more rapidly identify health effects. And we have at UCSF been experimenting with in vitro studies in this area, and that policy actions can be taken now to mitigate future harms. And with that, I will stop sharing and turn it over to Bart. Thank you.
Hey, welcome, Bart. If you can share your slides. Fantastic.
Yes, working on it.
Okay, great.
All right. Thank you for the kind introduction. Um, so, yeah, as a follow-up to Tracy's talk, I would like to dig in a bit more into the details of the diversity of plastic and what it means when you want to quantify or assess exposure. This is a picture of microplastics. Well, you see the diversity, I think that's clear. Um, and yeah, it's important to address the exposure not just as is, but in the context of what you want to use the data for. And yeah, in the next webinar, it will be about effects. And yeah, you can, for instance, assess exposure. Um, but yeah, the question is, how is the effect assessed? And do these two align in such a way that you can do a consistent risk characterization for instance, the different scenarios that Tracy already mentioned, for kids, for the elderly, for adults, people in a factory. Yeah, so it's important to see this as a whole at some point, because otherwise you would not know how to do the exposure assessment. And the same applies, of course, to the effect assessment. These two should be aligned. Now, this is again also illustrated in this slide. It starts with a problem definition. You need to know something, for instance, early life stage scenarios, and then you would define the exposure and the way you want to assess the effects. And you see this yellow arrow here in the middle of the slide, and that points to what we refer to as the toxicologically relevant metric. Because if it's about exposure, then for what should we actually quantify it? Eh? Is it the number of particles or the mass or the volume? And that depends on the effect mechanism. If you want to say something about effects or risks, different mechanisms could be at play, and each of them might have a different relevant metric. Now, if this is the case, then the exposure should also already take this into account. Now, and here you see a couple of examples: mass, surface area, volume, that according to earlier literature, yeah, just is thought to be relevant for effects in human organs and tissues. And then at the end of the day, this can be aligned and compared, and then you would have a risk assessment. So that is all meant to illustrate that, yeah, the stage of the exposure, it's important to already think about these things. Now, this is basically the same idea, but with another level of detail. You have at the left-hand side here, you see the sources of plastic products, and then we have wear and abrasion. It turns into small particles, it gets in the air, in the food, in the water. That's what we eat and drink, so that's the way we are exposed. And the relative magnitudes of those different fluxes from air or food, components of the human diet, is very important. And you might want to describe this via distributions. And then at the right-hand side here, that's the effects. This can be quantified in similar ways, also distributions, and then this can be compared. External exposure needs to be translated into internal exposure. Now, and I think you realize this is an active and complex and very detailed yeah, part of research at the moment to get this all connected. If you have that, then you can do quantitative risk assessments. Now, this webinar is, of course, basically about the left-hand side here. This slide was also showed by Tracy. It's from our recent paper. Yeah, and the point that I want to make here is that plastics actually can be seen as a continuum. Two particles are never the same, but when they come in thousands or 10,000s of particles or billions, then yeah, the characteristics can be described as a continuum. And we have also this continuum of yeah, of species, mouth openings. So the smaller the particles get, the more available they are for an increasing number of species, and also potentially able to penetrate tissues, especially when they are at approaching the nano size. So that means that in effect assessment and also exposure assessment, size plays a very important role. This is an overview of concentrations measured in different tissues and organs. Now that looks nice, eh? And so you are tempted to compare the numbers. Now that's a bit difficult because they're all measured with different methods. So the challenge here is that the data are not aligned. Different methods means that you cannot just compare the data. And the data often are not necessarily measured to meet the requirements for a risk assessment or exposure assessment in a regulatory context. So QA/QC screening of the data is very important. So two things are needed: QA/QC screening of this data, but also alignment of this data to make it all consistent. Now, this is just an example of where we are with these QA/QC aspects. For some criteria, like particle size or particle shape in studies, it's fairly okay. But for instance, for the verification of contamination, background levels, there's still a lot to win and to improve. Now, a maximum score is 2. That's the blue line here. And you see that this is for a number of studies. It's not yet ideal. Another problem actually is the huge volume of data, the number of studies. So how to keep track of this? Now, artificial intelligence tools may provide a solution here, because it is by now possible to automatically extract the relevant texts from studies and assess the quality and evaluate studies. Otherwise, this might take 4 or 5 years, and now it can be done in hours. So this is a big improvement. But these are also pilot studies, so there is also in these algorithms a lot of improvements to make the coming years, I would say. This is an example here at the left-hand side of concentrations measured in drinking water, so very relevant for human exposure. And you see that they're all measured at different size ranges. So there's just different studies, and those horizontal bars indicate the size range. Now, it's all very different. This can be aligned. Imagine that this yellow parenthesis is a part of the distribution that is measured. But if you know the distribution, so that would be the white line, the relative abundance of the particles as a function of size. So basically this says that small particles are more abundant. If you know the distribution and you measure only the yellow part, then you can extrapolate to the entire microplastic size range, for instance, from 1 to 5,000 micron. So this is an alignment that can be done and is a way to get exposure data with the same unit, all aligned to what could be default microplastic from 1 to 5,000 micrometer. Now, this can also be done for effect data. Now, that's not the topic of this webinar, but basically with some equations that take shape and size into account, you can translate the distribution or certain particle properties into other particle properties, such that the distributions that you get become consistent and then can be used for exposure assessments that make more sense than when you would use the fragmentary data. Here's an example where this is done for concentrations microplastic in bottled water. So it's a probability density plot. Concentrations are on the x-axis on the log scale. The original data, that's the gray bars. And so I think this is about 10 or 14 studies, 10, 14 bars, and then it's realigned with this mathematical calculation, and then you get the aligned data in blue here, so a bit higher, and then you can feed these data into exposure models to get a whole picture of what we are exposed to. Now, this can be done with external exposure models. This is an example of a study that we did a couple of years ago. So the left-hand side is a scheme for the exposure to plastic particles. We ingest them, they are biodistributed in the body, they are excreted. For all those processes, you can make calculations. The right-hand side part of this graph is about the chemicals that are associated with the particles. So these can also be modeled. Hopelessly complex models, I would say, but still a good start to understand what's happening. Complex, but also diverse particles, quite some uncertainties. And with these models, you can calculate the distribution of the concentrations that we are exposed to. And then you see that this is a wide distribution, very wide. And that's just the reality that we have. It depends on so many factors that exposure can be calculated, but the uncertainty is huge, many orders of magnitude on this horizontal axis.
Bart, we have 2 minutes, just giving you, oh, you're on time. Perfect. Thank you.
I'm on time. 2 minutes for 2 take-home messages. So yeah, the first point I hope I made clearly is that exposure and exposure assessment, that's very important. But yeah, it should be done in the context of effect and risk assessment ideally. So a framework should be designed beforehand, a priori, so that you know what you need to assess when you assess exposure, which metrics, which units, etc. And the good news is there are tools available for aligning data and for determining data quality. However, the outcome of these quality assessments, unfortunately, is that the data often is not yet of perfect quality. So there is a lot to measure and a lot to do. Thank you.
And Tracy, fantastic. A lot of information in a very short time. Now we have about 15 minutes for questions, and I'm going to try and speed through some and also ask a few from the Slido. So first, let's go back to Tracy for a second. And Bart touched on this, but there are chemicals in plastic as well as the polymers themselves. And so can biomarkers of microplastic exposure be identified?
So I want to say, yes, so there's microplastics themselves, which Bart talked about and I talked about, and that have been measured in multiple studies, but there's also chemicals that are in the plastic, and some of them we already know a lot about. So phthalates, for example, which are found in, are used in plastics often to make it flexible, bisphenol A, and then there's additives like flame retardants can be added, colorants, additives. So we know a lot about a very small percentage of the many different plastic chemicals that are used in plastics. So EPA, there's been some publications about the number of chemicals used in plastics. It's about, we're actually doing a little bit of studying on those 10,000, and Bart can correct me if that's, it's because it changes all the time, the number. And we've probably measured about 100 of them in people. So there's a lot to be gained to understand the extent to which these different types of chemical, and that's just the chemicals that we know that are put into the plastic. The chemicals also can break down or they can interact together to create other chemicals. So I think there's a lot to be learned here. We've been using non-targeted analysis, high mass spectrometry non-targeted analysis to try and scan for chemical signatures in this plastic set of 10,000 chemicals and are finding more that are traditionally monitored. But we do know that some chemicals that are in these plastics are measured in people. And there's more to learn about that because there's a lot that haven't been attempted to be measured.
Thanks. Bart, also, this is something Tracy talked a little bit about, but and it also relates to where do these plastics come from and what polymers are they? Can you talk a little bit more about how the degradation of larger plastic debris, which we're more familiar with, contributes to the formation of microplastics in various ecosystems?
How it contributes, yeah. I think you could refer to big plastic or macroplastic as young microplastic, right? I mean, the source of microplastic is largely by degradation, fragmentation, abrasion, wear of microplastic items. They break apart, become smaller. That's basically where microplastic comes from. There's a small fraction, which is primary microplastic. It's just as is. The particles are in a product already small. But calculations have shown that the major part comes from fragmentation, abrasion, wear, breakup of particles.
Okay, well, we have a question on Slido that's very specific, so I thought I'd ask that. And I'll ask it of both of you to see if it applies to you. Have you studied how plastic in gardening, composting, and farming impact human health? This person said they're concerned that plastics are impacting those things like in our community.
Well, I think the first part of the question is the extent to which, so farming is pretty broad. It's like, so I mean, I think what we've seen from studies, and Bart can also answer this, is that microplastics are ubiquitously transported around in the environment. So there would be microplastics in soil, there would be microplastics in air. The extent to which farming practices generate more microplastics, I don't know that there's really an answer, but I think what we've seen is that people are exposed to, I mean, people are tissues that have been collected and measured find microplastics. But I would say that probably we don't know a lot about farming practices and microplastics, but maybe Bart will correct me on this.
Well, I agree. Literally, is there anything known about effects of farming practices and microplastics on human health? I think there is no study that has that in the title or the abstract. But yeah, some more indirect things can be said. And in that sense, I simply agree with Tracy. Yeah, there are some practices in agriculture that like plastic mulching, and okay, and those foils
Right.
slowly degrade, and then yeah, you have microplastic.
Yeah.
There's polymer coated slow release fertilizers, which is basically a small plastic sphere with the nutrients in it, and then after some time it falls apart, it fragments. So in all those contexts, also in a farm, yeah, plastic is used, and yeah, and on top of that, there's the flux from the atmosphere, atmospheric deposition, and background contamination. So yeah, there will be exposure also there.
I just want to say too, the other thing that I think is important, because there's some practices now to take a wastewater treatment plant waste, which that will should probably have microplastics in it, right? Because people are excreting microplastics, and then that's going to get into the waste. And some of that waste product is being considered to be used, maybe not in agricultural lands, but maybe in landscaping areas. And so that could be reintroducing microplastics back into the environment. So I think actually the state of California has been focused on trying to look at microplastics in water sources and drinking water and these types of activities. And I think that's a really important area of trying to determine how to, basically remove it.
Great. Well, getting a little to the heart of this, I thought I'd follow up with you, Tracy, on this question. We've sort of said exposure is a component of risk and probably a large one. And so what have you been seeing as a physician over the last several years about microplastics and acute and chronic exposure?
Well, I'm just going to say I'm not actually an MD doctor, but that's okay. I like to all the information we're doing, we inform our MD colleagues. So I think what's been interesting or important or concerning is that some of the studies that have looked at microplastic measurements in people some of them have looked at microplastic measurements over time. So they've looked at levels, over 8 years or 10 years, and finding that the levels are increasing. I know there's a lot of debate about some of the methods, but the overall part is that we're seeing increasing exposures, which would be expected because plastic production is increasing. And as I said, anytime you see an increase in emissions of any kind of pollutant, exposures will increase. And then as I think there's going to be a future webinar on this, but also something that we did as part of the report for the state of California and subsequently published was do a systematic review on some of the health effects that could be related to microplastic, including finding suspected links to reproductive, digestive or colon cancer and lung effects. And those are, I think one of the concerns that people have is that some of these, and we've getting these requests at UCSF ourselves, is people are seeing increasing colon cancer among young people. And so clinicians are concerned about what's the role of these increasing exposures to chemicals, including microplastics. So I think that's why this is a really timely topic for this series.
Thanks, Tracy. Bart, this is a popular question on Slido also, but what are the critical data gaps in linking microplastic exposure, which is what we just talked about, to human health outcomes?
Yeah, that's a big question. So I can give some answers, and then after an hour of thinking, I might give some more answers probably, but we do not have that hour. But I think that, yeah, the difficult thing is we can calculate how much we inhale or ingest, but then what? So which fraction of the particles is actually transferring or penetrating cell layers, and how is that distributed in the body? So if there is an effect somewhere in a target site in the body, how do the particles get there? Are they bioavailable? How long does it take? How do they stay there? So yeah, basically the translation from external exposure, the biodistribution, and then the point that they get at a certain target site. I think that is a big data gap to be able to, that we need to answer, to be able to translate future emission levels into increasing concentrations in humans. So that's more prospective thinking. You could also say we just measure in tissues and organs. That's of course for ethical reasons difficult, but there may be possibilities with post-mortem studies or other ways to just see what's there. I think that's what Tracy was referring at also with the mechanisms that she described. And that is very important. But to be a bit more predictive, we would need this information about external to internal exposure, I think.
Thanks. Well, going into the nub of this, which is maybe Tracy, you could start, and Bart, you could circle back, which is what data would be needed to inform regulatory standards, unless, and there are many, of course, and how can surveillance programs better support policy development? How do we get there?
Well, I think there's different ways to think about policy and regulatory programs, because we know already from lots of experience working with understanding about just chemical in general, or I like to think about air pollution. So air pollution, we have a lot of experience in the regulatory community in the United States around particulate matter air pollution. And the thing, and as Bart was saying about respiratory related air pollution, is that microplastics really are part of the particulate matter 2.5 group. And so I think that so you can use that as an analogy in terms of thinking about when you're looking at different regulatory areas that are focused on certain types of exposures, for example, air pollution. That's a really good opportunity for thinking about, well, what's the role of how is microplastics part of that? And regulatory activities we focus on in terms of particulate matter air pollution and reductions and contributions to that will also reduce microplastics. So efforts to lower effect. Well, the last administration recommended lowering the particulate matter 2.5 standard to lower levels that would therefore have to reduce microplastic contributions to that. And I think that also it's really important because we know already that people are exposed to microplastics. It's true that our health effects understanding is what we've classified in the suspected range, but because there's going to be increasing exposures, it's very important for the government to be thinking about because what ends up happening, and I think this is a really important point that come up in the panel, is these are persistent and bioaccumulative. So if we don't make the right choice now, we're going to end up with a problem that will be very difficult to correct. And we see this consistently with whether it's DDT or PFAS chemicals we still measure DDT in people today. So, and that was banned many years ago. So what we have to do is think about, we should be lowering, because if it's going to be tripling, maybe we should be thinking about capping it so that we don't have, wait until we get a lot of exposures and then oh, wow, we really know now the health effects rather than we're just suspected on the health effects. I think that's an important component of thinking about policy activities. I gotta say more about this, but I know we are limited on time. So I don't know if Bart has anything to add.
Well, maybe a small comment that the precautionary approach, I think, is very good. I think it's basically also what you said, Tracy. We can spend years or a decade on understanding everything, all the details. I think a precautionary approach is very important based on reading across what you mentioned, air pollution, see whether some policy approaches can be deduced from read across approaches, while at the same time moving on with research to get a more refined view. So do the one thing and not forget about the other. Also refined, more detailed research to get answers, because that will be important to be able to design other polymers or products in another way, or set priorities on where to act first. Resources are limited, so a more refined risk assessment method is always useful for prioritization, I would say.
Yeah, agreed.
Okay, I'm on my last question now, and it's hard to choose. I'm looking at the Slido questions. Let's see. Well, let's go back to, I think, Bart, you said size matters. Essentially, that's what we have heard anyway. And so what is a critical size for microplastics to be transmitted from the human digestive system into the rest of the body? And what factors control this process? It's very...
I'm not a toxicologist and not a medical doctor, but I do know that there is a lot of biological variability. So that people might say 10 micron or 1 micron. I think we might be surprised. So I think those boundaries are important, and they are used in models, but we should give some distribution. And we also maybe tended to think about the healthy body first. But there are also many situations where somebody is already ill, or there's a certain syndrome, and then there are already some things going on, and then plastic particles on top of that could give the fatal push maybe. So maybe we should focus on people that have a certain disease first. So I'm just brainstorming now, but 1 or 10 micron are often mentioned, but it would surprise me if there would not be also sometimes some bigger particles might pass occasionally.
Okay, great. Tracy, do you want to follow up on that?
Well, I think that's a really excellent answer, but I just also want to say there's still also what we call point of entry contact effects. So if you're breathing it into your lungs, maybe it's not absorbed, but it can still damage your lungs, right? So I think that, or if you're eating it, it goes into your gut, it could still be because it can cause what we've seen in studies that have been looking at this, for example, inflammatory responses, for examples. It could be because it's just being aggravated by these, even if they're larger particles. And I just also want to point out that your original question, which was the biomarkers, I mean, there's also, and we don't really know how much of this is from the particles themselves or the chemicals that are coming off of the particles, but there's certainly chemicals that can come off of these particles, and those we know can travel around the body.
I'm going to follow up with maybe a short answer I can get from Tracy, because this is a very specific question. Health authorities often recommend boiling infant feeding bottles to sanitize them. Is there any guidance on microplastic release? And this would be a short answer, I think, for you. You might have any.
I would use a glass bottle myself. So I'm just going to give the answer from my own personal, not that I haven't also used plastic bottles, but I think that heating releases chemicals and plastics. So I think that's a concern for your infant.
Okay, thanks for that extra squeezed in question, which seemed very personal to some of our audience. So thank you very much. I think we've now hit our mark to transition to the next session. And I really want to thank you, Tracy and Bart, for really diving into this topic in a very quick and effective way. Appreciate it so much. So we're going to transition to session 2, which will be moderated by Dr. Daryl Boverhoff, who's the Sustainability Director with Dow, and he's also a member of our committee. And so I'm going to turn this over to Daryl. Thank you very much.
Great, thanks, Margaret. So good day to all. My name is Daryl Boverhoff, and as Margaret mentioned, I am a Sustainability Director with Dow. And for the past several years, I was a member of the National Academy's Environmental Health Matters Initiative, or EHMI, and I'm also one of the planning committee members for this webinar series. So the goal now in this next session here is to have a panel discussion that's really going to focus maybe a little bit more detailed on some of the questions that are coming in the Slido, but also to highlight the successes we've had in studying microplastics, as well as the lessons learned and what these research experiences have taught us that we can apply towards future work. We also hope to highlight where progress is occurring and where there are still gaps that will help to inform our next steps in exploring the assessment of the impact of microplastics on human health. On the panel, we will have our two speakers that you just heard from, Tracy and Bart, but they will also be joined by two additional panelists. And we're going to give each of those panelists a chance to have 3 short-minute introductions that will cover a brief overview of their background, some initial thoughts on where they see opportunities in understanding microplastics, and then this will be followed by questions from the audience with that full panel. So just as a reminder, the biographies for all of our speakers and panelists can be found in the agenda and on our website. And our first panelist is Dr. Jane Muka, who is the Managing Director and Chief Scientific Officer at the Food Packaging Forum. So Jane, can you please turn on your camera and unmute? And we'll give you a few minutes to provide your opening remarks.
Wonderful. Thank you so much. Hello, everyone. It's a pleasure to be part of this important webinar. So I focus on food contact materials, which is food packaging, but it's also food processing equipment, anything that is purposefully brought into contact with food. And I'm interested in understanding the health impacts of the chemicals that can transfer out of these materials and that people ingest when they ingest food. And more recently, we've also started looking at micro and nanoplastics, which are generated from the normal and intended use of food packaging and other food contact materials. And I actually don't have a lot to add, to be honest, because I think Bart and Tracy did a phenomenal job. But I do want to emphasize a few points that have been made already. Tracy mentioned that plastic chemicals have been measured in people. We've actually systematically mapped the evidence on that for chemicals that leach from plastic food contact materials, so food packaging, for example. And it's 1,396 chemicals, so 1,396 plastic chemicals for which we have evidence that they've been measured in people. And we also know that they are either present in food contact materials or that they are used to make food contact materials. For the chemicals that we know that transfer from food packaging, plastic food packaging into food, we also have another data set which is publicly available, and I'll provide the links to those, where we have evidence now, empirical evidence for 1,481 plastic food contact chemicals that they actually migrate from the plastic food contact material into food, which makes human exposure very, very likely. What we've started to work on now, and I'm sad this isn't published yet, but I hope it's going to be published in the next couple of weeks, is a similar systematic evidence map for micro and nanoplastics that are generated from the normal and intended use of food contact plastics. This field is only just emerging. So there aren't a lot of studies where we have high confidence that the quality assurance, quality control mentioned by Bart before has been done properly, and also where the study design actually allows conclusions about causality, that the microplastics found in food actually originated from the food packaging. But there are a few. There are a few such studies. So there is emerging evidence. And I think this is also a really important point to make. The food contamination with plastic chemicals and micro nanoplastics comes directly from the packaging and from the food contact materials. It is not only that plastic waste breaks down in the environment and then gets into the food chain. It's actually probably a lot more closer to the food. So I think that's an important lesson to learn here, because it tells us that if we reduce our kind of dependence on plastic packaging, we can most likely also reduce exposure to micro nanoplastics and most definitely reduce exposure to plastic chemicals. I know that this issue may sound complicated if you're new to this, but it's actually quite simple. I think, as Tracy said it before, if we reduce plastic in contact with food, we will reduce human exposure to micro nanoplastics and the plastic chemicals. And with that, I think my time is up. Back to you, Daryl. Thanks.
Perfect. Thanks, Jane, for your opening remarks. So we'll turn it over to our next panelist, Dr. Matthew McLeod, who is a professor of environmental science at Stockholm University. So Matthew, can you please turn on your camera and unmute, and you'll have 3 minutes for your opening remarks.
Thank you. Disabled my camera, Daryl. It says my camera is disabled by the host. Oh, now I'm back. Very good. Yeah, I prepared a few slides. I guess you can see the picture, Daryl?
Yep, we can see it.
Okay. Yeah, I'm a professor of environmental science at Stockholm University. I'm mostly interested in how plastic behaves in the environment, at understanding how emissions move through the environment, how this leads to environmental exposure and also human exposure. And Bart said in his opening comments that macroplastic is just big pieces of plastic are just plastic that's waiting to turn into microplastic. And this picture I put on this introductory slide is a power bar that's in a conference room just down the hall from where I'm sitting now here at Stockholm University. And this thing has been sitting in the sunlight next to the window for a few years. And when you pick it up and just put a little bit of pressure on the edges, it just fragments into microplastics. So I took a picture of that just as an illustration of the potential for plastic to turn into microplastic. And what I want to say actually is going to reinforce some of what we've already heard, especially from Tracy in her comments. The general topic today is microplastics in the environment and human exposure pathways. And if you draw a really simple picture of that, that's this. We have an environment, an exposure environment. We have people living in that environment and plastic coming in. And this is something, exposure to environmental pollution is something I've been thinking about my whole professional career. You can see this is a figure that I adapted from a paper from over 20 years ago that I did together with my postdoctoral supervisor, Tom McCone, at the Lawrence Berkeley Lab. And one of the things that Tom worked on back then was formalizing how we think about intake by a population living in an exposure environment into really simple equations that give the boundary conditions for why this happens. And they wrote this nice paper in Environmental Science and Technology that argued that a good way to think about this is that intake at the population level is just directly proportional to emissions to a pollutant into that exposure environment. And I think that's the first thing that Tracy has also been saying, that emissions are something, the total amount of plastic that we're putting out into the environment is something that we have to consider when we think about total population intake. But what's this proportionality constant? They called this the intake fraction. And together with Tom, I worked on decomposing that intake fraction into its component bits. And so that proportionality constant can actually be thought of as a competition between two processes. The first one is represented by all these arrows that show the plastic leaving the exposure environment. And that you can think of as the residence time of plastic in the exposure environment. If you put plastic into the environment and it's quickly removed, there's less chance for the population to be exposed. But that process of removal is in constant competition with the plastic finding its way to a member of the population and causing exposure or causing intake. And that other denominator there is what we call the characteristic time for intake. And built into this denominator is all this stuff about exposure pathways and how efficiently the plastic moves through the environment to us. So I want to just take a step back and think about what we know about plastic. We know that emissions are increasing exponentially, and Tracy highlighted this, and that intake is directly proportional to those emissions. We know that the residence time of plastic in our exposure environments, it's variable, but it can be very long, and it's often very long. We're talking years to decades, and this is very long when we think about other pollutants that we have experience with. And then finally, this characteristic time for intake, this one's quite uncertain and highly variable. And I think Bart showed this in one of his slides, that you can have very high variability between people in terms of how much plastic they're exposed to. So this is uncertain, but we have two of these things that are blaringly obviously concerning if we're concerned about population level intake of plastic. And I think that that's where we can focus our attention actually, is on the increasing emissions of plastic into our exposure environment and the fact that it is has very long residence times, very long persistence in our exposure environment. And this is the kind of thing that I hope we can talk about a bit more in the discussion that's coming ahead.
Perfect. Thanks, Matthew. Really appreciate you sharing that intro, as well as Jane, your words. So with that, we're going to kick off the full panel. So I'm going to invite Tracy, Bart, and Jane all back to the screen to join Matthew and I as we begin the Q&A session. And then for our audience, keep submitting your questions through the Slido. Keep upvoting the ones that you'd like to see answered. There's a lot going on behind the scenes here. We're going to try to prioritize those questions to make sure we get your questions answered while we also have a good discussion. So I'm going to go into the questions now. And Matthew, I really like the diagram that you showed in terms of the different compartments that exist that can contribute to sources of human exposure. So based on your assessments, where do you really see the highest concentrations of microplastics in the environment? Is it air? Is it water? Is it soil? What are your perspectives on that?
Well, the short answer here is that the highest concentrations of plastic and microplastic in the environment are close to sources. So if you go into urban areas are higher than background areas, go close to a landfill and you'll find lots of plastic. This relationship exists right down to the micro scale. Go close to the litter box, to the bin where people are putting plastic in, and people will miss, and you'll have higher concentrations of plastic close to the bin. So closer to sources is where you have the highest concentrations. Generally, they're going to be falling approximately exponentially as you move away from those source areas. Plastics are not volatile, so you don't find high concentrations in air, but that doesn't mean that the concentrations that are in air are negligible. That's still an important pathway for plastics to be mobilized and moved through the environment. And the other point I want to make about this is that plastic in the environment is at much higher concentration in developing countries than it is in developed countries. I'm doing a little bit of work together with some colleagues in Africa, and when you travel to Africa and you walk around in urban areas or in near waste disposal sites, it's shocking how much plastic you see just laying around. So that's the general picture that I would want to give to that.
Okay, perfect. So maybe kind of building from that. So Matthew talked a little bit about, exposure being dictated on proximity to the sources in terms of those environmental components around air, water, soil. So Jane, for you, obviously you're with the food packaging group there. From your perspective, what are the kind of main sources of exposure and the exposure routes for human exposure?
So thanks for that question, Daryl. Yes, at the Food Packaging Forum, which is a charitable foundation, we're an independent research organization. We're mostly focused on, as the name says, food packaging and food contact materials. And I think I explained that quite extensively in my introductory comments. In my point of view, food packaging as a source for direct human exposure to micro nanoplastics is underappreciated right now. So it's not actually, there's actually not a legal requirement for those who put these products on the market to assess the generation of micro nanoplastics from the normal and intended use of these products. So I think that's an interesting space to explore and to generate new knowledge and data on. Other sources, as Matt said already, you know, tires. Tires have probably been best documented for many years. People assumed that exposure, airborne exposure to tire particles and other plastic particles intake through the lungs is one of the key sources. Paint, of course, there's so much paint everywhere and it ages, becomes brittle, but doesn't degrade chemically. That's another source. And then we do have some data showing contamination of foods, seafood, shellfish, for example, filter feeders that suck in microplastics with water, and then the plastics stay in them, and so on. So I think Matt gave a really good answer. The sources of microplastics are where we use plastics.
Perfect. Thank you for that. We're seeing a lot of questions in the Slido that really deal with the kinetics of microplastics. So how they're moving in the human body, how they might be moving through the environment. So Bart, you had a picture in your presentation of the human body with potential exposure, potential areas where they've been found in tissues. So how are researchers trying to figure out how much of microplastics that are ingested actually stay in the body and how much are excreted through the urine and feces? I'm sure there's been some isotope labeling or fluorescent labeling done that maybe has looked at the ADME principles of these things. Can you comment on that?
Yeah, definitely. Thanks for that question. Yeah, this is the domain of PBK modeling and parameterizing these models, and that's a lot of work. The models are complex, a lot of uncertainty. There's quite some work done on rodents. So the kinetics of microplastic is best understood for the mouse, I would say, probably. Quite some studies. PBK models are made and then calibrated and validated for these animals. That works reasonably well. And then you can calculate the fluxes, predict the concentrations. It's still not much, so maybe a handful of studies. The big question is how to translate that to the human body. There are relationships, allometric relationships that you can use to translate, but the validation is still not there for the human body. There's a lot of data on concentrations, but they come from different studies, right? So one study focuses maybe on the brain, another one on the liver, another one. So it's never one body which is looked at. So what you mentioned, like radio labeled studies, I think they are being done now in the context of several projects. So I'm very optimistic that we are hopeful, that's a better word, that we will see some of those results in the near future so that we can calibrate and validate those models much better.
Perfect. So the corollary to that with respect to the pharmacokinetics in after human exposure, and this kind of links back to the gardening question we had earlier, but a little bit more specific. And I'll leave this one open to anyone who wants to tackle it. Has there been any documented evidence on microplastics in soil being absorbed or taken up by plants, agricultural crops, so that vegetation component that Matthew had in his slide, grown in contaminated soil? So, plants are different. They have a cell wall versus, mammalian cells. So has there been any studies on that? Again, open to anyone.
Maybe I can just jump in here quickly. And I'm not an expert on this, but I have seen studies on this. Actually, I'm part of the European Commission-funded research project cluster, that's a mouthful, researching the health impacts of micro nanoplastics. And we had a meeting 2 weeks ago in Brussels where we presented kind of after 4 years the research findings. And there some colleagues did present findings on that. So they looked at the uptake into basil in this case. And I was interested to see that the microplastics kind of seem to cluster in the stems, not so much in the leaves. But again, it's very kind of unrealistic lab-based conditions with, I forget what exactly the microplastics were that they use, but in the environment, things are a little bit more complex. But we have seen that. And let me just add here briefly, and Matt can maybe come in on this, but microplastics are found in sewage sludge. And they get in there, for example, from washing of polyester clothing, which is a lot of clothing these days. And polyester is basically PET. The density of PET is larger than 1. So in the sort of settling basins of wastewater treatment plants, polyethylene will sink to the ground and it will settle in the sludge. And many countries in the world actually still use sewage sludge to fertilize crops. And so that's a kind of direct, unfortunate in a way, exposure source of spreading these particles in soils. And I do think that may be a practice that requires a little bit of rethinking.
Yeah, if I could just, since you directed it to me, Jane, I could follow up that I think this is something that we should really be concerned about because we have this recent experience with the perfluoroalkyl substances, which are also accumulate in sewage sludge. And when you take that sludge and apply it to agricultural land, you can render your agricultural land irreversibly, for all practical purposes, contaminated. And I think that is the case with perfluoroalkyl substances like PFOS and PFOA. And I think I see no reason that won't also be the case with micro and nanoplastics, because they're not going to be very mobile in these agricultural soils. And the extent that they're bioavailable to plants, I think, is an open research question right now, but we shouldn't just assume that they're not going to be bioavailable because actually our experience with these kind of things tells us the opposite is probably true.
Perfect. Hey, good responses, good things to think about, not only in terms of potential for exposure, but some of our practices and how that might influence exposure. So I want to pivot to a different question now that's getting through in the Slido. It's, what do you think about FDA's opinion recently that there's not sufficient scientific evidence to show that microplastics and nanoplastics from plastic food packaging migrate into food and beverages? And Jane, I know you probably want to speak on this one, but let's maybe first hear from Tracy.
What's the date that this was put out? Well, let me just say, I think that it's kind of challenging because it depends on the regulatory bar that's being required of these agencies for making decisions. But I think we've also seen that some government agencies haven't used the most up-to-date science in their decision making. FDA also was considering a petition to ban phthalates in food packaging. So phthalates are used in plastics. We've talked about that you can not only be exposed from food packaging to microplastic, but also the chemicals that are in these plastics, which include phthalates. And there's well-established evidence that phthalates can increase risk of numerous adverse health consequences. Unfortunately, in that petition, FDA did not actually consider all the latest scientific information. It's hard to say what the reason was, but they denied that petition. So I think part of the challenge is, government agencies can be a little slower in their uptake of scientific methods, and we really want to see them accelerate that. I can't speak to what's going to happen now, but the current administration has indicated that they want to relook at some of the rules about how FDA evaluates contaminants that are allowed in food, and which I assume will also include plastics. So maybe Jane has some insights here since she works on this so extensively So it's a moving target right now. So
Hey, so what I wanna say here is that FDA has a tough job, hey?
yeah.
It's really not so easy to determine whether there is a risk or not when you have such huge data gaps. So if we just look at that from that perspective, I would agree that with the current data availability, it's actually not possible to determine if there actually is a risk or not. We talked about the precautionary principle. And in the absence of robust science, it really becomes a values decision whether you say, okay, I can't say there's no harm, so I'm assuming there is harm. And I personally assume that there is harm. I lean towards the precautionary principle because that's my set of values as a human being. But from a very strictly scientific perspective, I think the current data set will not allow us to say there's a risk or there's no risk, right? Now it becomes a political discussion to say, should FDA have put out a kind of opinion saying there's no risk of micro nanoplastics when they actually can't make that conclusion scientifically? That's a different matter, right? I do want to point out that there was a recent government accountability oversight report on the way that FDA enforces its rules on food contact materials. And that report published in October 23, I think, maybe it was 22, don't hold me accountable for that. That found that there are risks associated with the way FDA does it. And those risks pertain to chemicals. So there's definitely room for improvement in the way that FDA is laying out the rules and enforcing them for food contact materials. But when it comes to micro nanoplastics, I think the jury is still out. However, let me just finish that out. But I think two things. First of all, people that are concerned about plastic and health impacts, either from chemicals or from micro nanoplastics, they do well by avoiding plastic packaging, and they do well by avoiding heavily processed foods, because the processing will also introduce those chemicals and micro nanoplastics. The other point is, I think we need our regulators to have mandatory testing for the generation of micro nanoplastics from the normal intended use of food contact materials. I think that's a regulatory spot. And once we have that regulatory requirement, we will be able to fill the data gaps. And then hopefully we will have enough data to do robust risk assessments.
Yeah, I agree that the regulatory program right now for chemicals on the market and microplastics does not require that companies provide the data either to tell us where they're used or to provide the data so that we can assess the risk. But I would say, based on our own evidence evaluation using gold standard methods, that it is definitely suspected. And to me, that's in the government, having served at EPA, EPA can make decisions, or government agencies can make decisions on things that where the evidence is uncertain, and you do want them to do that because people are being exposed right now. So if we wait for 100% certainty, we also will continue exposures that we already suspect are causing health harm. So
Yep.
I think those are challenging things in the regulatory program. There's a lot of pressure. I totally agree. There's a lot of pressures when you work in the government about making decisions, 100%. But
Yep.
they're also charged with protecting the public. So.
Oh, perfect. Good thoughts. And certainly agree. I mean, the regulators right now have a tough job based on many things, including addressing the safety of microplastics. So I do want to pivot to another question. It's getting upvoted in the Slido, or so I'm told. That's related to certified biodegradable compostable plastics that could reduce exposure to microplastics and perhaps then provide an alternative. One of the things I know, there's different alternative assessment programs out there that consider concepts of substitution and regrettable substitution. So has anyone thought or looked at these things in terms of biodegradable compostable plastics as a potential solution?
Have you looked at this specifically, Bart? Because I could offer my thoughts here.
Go ahead.
I don't consider myself an expert on all of these biodegradable / compostable alternatives, but I have looked into them a little bit. And the ones that I've looked into, they are compostable in industrial compost systems, which I think is an important point. It doesn't mean that you can put them in your own backyard compost heap and expect them to be degradable. They need industrial compost conditions, which are quite higher temperatures and much more efficient than typical compost systems. And then at least some of them, they are biodegradable into microplastics and nanoplastics faster than, and not biodegradable into carbon dioxide and terminal products. So in that sense, they're not a total solution to the question, because they're not biodegradable into something that isn't a micro or nanoplastic. So the ones that I've looked at, I don't see the current ones as great solutions. I think there is opportunities here. I think that this is something that we should be looking to in the future, which is design of materials, green design of materials that have better properties for the environment, as well as that are fit for purpose for application. And then the question goes on to ask about health impacts from this, and I think that this is just a complete unknown. To speculate about whether these alternatives have lower health impacts when we have such high uncertainties about health impacts to begin with, I think would just be not something I would want to do.
Are any additional perspectives on that? I know Matt kind of called you out there.
Maybe a small comment that it's often said, then, is this a solution? I think there are not really single solutions. So it's often said, and I very much agree, that there is no silver bullet solution to the plastic pollution problem, that it has to come from a wider range of things, design products in a different way, other materials, less plastics, avoid coming in contact with, use more glass than plastic. However, you should do a cost-benefit analysis, an LCA, and LCIA to see whether these alternatives are really fully better. But I very much agree with what Matt mentioned about that you can have a burst of exposure for those materials in certain circumstances. I think that would be then specifically relevant for ecological systems. Maybe a human would be a bit further away from such a setting on that time scale, but still, that's also worth considering, of course, the ecological damage that you could have.
Jane, maybe a quick response to this before we go to the next question.
Just 2 quick points. First of all, I just want to mention that when it comes to the chemicals and the hazardous chemicals, bioplastics are not better than plastics, right? Just because it's bioplastic, it has nothing to do with the kind of chemical composition and potential leaching of hazardous chemicals. And there are a couple of studies showing that there are hazardous chemicals leaching out. Second point is that, at least where I live in Switzerland, composting facilities don't like bioplastics. And the reason is because people, consumers, citizens, can't distinguish easily between those plastics that degrade in industrial composting and those that don't. And so they get a lot of contamination from non-degrading plastics because there's confusion. So they prefer not to have any plastics at all.
So super interesting perspective there. And I'm wondering if that's a point when we get to our 4th webinar on risk communication and other things about solutions, that might be something to make clear, because marketing claims can often lead people to maybe make solutions that aren't really a solution. So I'm going to go to the last question, and I'm going to ask people to be efficient in your response, so maybe around 30 seconds or less. And it's kind of a high level thought question. So, and I'm going to start with Matthew, and then Tracy, and then Bart, and then Jane, simply because that's the way you guys are ordered on my screen right now. So the question is, when it comes to microplastics and improving our understanding of their distribution in the environment and human exposure potential, what do you see as the most critical need?
Ooh, that's not the question I thought you were going to ask.
Oh,
What do we see as the most critical need for improving our understanding?
Well, you use up your 30 seconds.
Yeah, I'm using up
You
my
want me,
my
I
30
can say
seconds. Thank you.
something while you're thinking.
Go
if
ahead,
you'd
Tracy,
like.
you go ahead.
I think what in this case, because of course we were thinking you might ask something else, but I'll just say I feel like a good government repository that is accessible, for example, that that researchers can put their data in and that we can access and then mine, I think is going to help really accelerate this. A lot of all these studies are all disparately dispersed and it's hard to aggregate it. I think that, like, for example, the EPA has a chemical dashboard. They keep aggregate information about all the chemicals that are used on different lists. That's what we could use for microplastics. I'm going to say and that will help be foundational for research.
Right.
My turn.
Sure.
My vote goes to measurement of the smallest fraction, below 10 micron, below 1 micron. So yeah, basically nanoplastic. It's there, but it's still quite a hypothesis actually. We need to measure it if it's there and what it does. So yeah, microplastics and human health implications, I think it's crucial that we can measure small particles in a wide array of relevant matrices.
Perfect. Jane?
I thought Matt was going to go, but I'm happy to jump in there. I like what Tracy and Bart said. I would be in favor of both of those. Maybe in addition, I'm looking forward to having data sets of large cohorts, like big population groups. And we're actually working on one of those with the Aurora project funded by the European Commission, where we have 700 mother-child pairs. We have blood samples, urine samples, placenta samples. And we are assessing them for presence of micro nanoplastics. And so I look forward to seeing what my colleagues, the epidemiologists, will do with these data sets. And that should be published within the next year or two.
Perfect. And full circle back to Matt.
Yeah, finally, thank you. I endorse what everybody said. I think Bart's point about measuring is good. And then Jane stole my answer after I thought about it, because I really think epidemiological studies are super important for demonstrate, like, if your bar is going to be that you have to demonstrate risk, I think experience tells us that for these persistent chemicals or for persistent pollutants where you have population level exposure and no control group, the way to discover effects at the population level is with epidemiological studies. And again, this is from PFAS and PFOA experience, where a bunch of associations showed up once you did these epidemiological studies. So I think, in a way, I didn't want to say this right away because this is reactionary, and I would like to be proactive. But if you're going to demand risk information before you make a decision because you don't want to act based on precaution, then I think epidemiological studies are the quickest way to get there.
Awesome. Hey, I think that was a great kind of summary in terms of, looking forward in terms of what we need. Really appreciate the input from all of the panelists. Very insightful. Obviously, there's certain things we know. There's certain things we don't know. Regulators have a tough job, but let's continue to collaborate, share information, make sure we design really solid studies, including epi studies, so that we can make some additional conclusions about exposure and then ultimately risk. So really great panel. Appreciate everyone. And I'm going to turn it over to Margaret to take us home.
Great. Thanks to everyone. This is I think we would all agree we'd love to have all of you for coffee and talk about this more, because you all are incredibly aligned on many topics. One thing I just wanted to say was I want to say I think this was a great discussion. Let's talk about what we covered a little bit. So, and I encourage people to rewatch this when it will be recorded, because there's a lot of content here. We heard that increasing plastic means there's increasing microplastic. So production is linked to microplastics. We talked about top sources, and one of them was packaging that was discussed at length. And also, though, that it's in all the compartments, and they're connected, whether it's air, soil, water, and there are certain practices that may be exacerbating this. The food packaging piece was brought up in particular. That chemicals are one source of toxicity, or potential toxicity in plastics, but also there's physical impacts from microplastics. And so those are two things to look at. And that there's exposure differentials that were really important to talk about, which is children and infants, developing countries. And all those already who are ill might have, we might want to look at more. And that those living near sources of plastic, which is where a lot of the microplastics live, is something we should be focusing on. We also talked about the value of taking a precautionary approach. We understand regulatory limits are one thing, but until a regulatory limit is reached legally, it's important to not only flag suspected impacts, but also take actions to reduce exposure. And there were a few thoughts there. So I just wanted to flag those, that avoiding exposure is one thing that you can take from this. And there's some areas that we talked about. The risk and the health impacts will be our next webinar. Also, thank you for the data gap discussion. I think that was great. And there's more to do there. So as we wrap up, first of all, thank you to this panel. It's fantastic. The next webinar, as I said, will be on potential hazards to human health and the complexity of assessing risks, which we really touched on here. And so we're building an appetite for that webinar. And if you want to stay informed for these future webinars and our activities under this EHMI group, please subscribe to the Environmental Health Matters newsletter on our website. And finally, your own voice matters. Please take a few minutes to fill out this post-meeting questionnaire that will continue to inform the series. Thank you again, Tracy, Bart, Jane, and Matthew for a really stimulating discussion. Thank you, Daryl, too, for your help in moderating this. We hope to see you all in April, and the webinar is now adjourned. Thank you.